Oh, right now it's 1 hour class.
Yeah.
Okay, I have gotten the notification like it'll like 1.5 hours they said. Okay, fine, you can switch, like keep your videos off. What is the last thing you wrote in the previous class? This line only, right? Third line.
Yeah, yeah, yeah. RER and then right pass on to Golgi.
Okay, so we had completed Golgi apparatus and rough endoplasmic reticulum, right?
Yes, ma'am.
Okay, so today we'll do one topic only, and the rest things you can write. So we'll do lysosomes today. Okay. Now talking about lysosomes, so these are the tiny, simple, spherical sac-like structures. Now these are those structures that are formed as a result of ER and Golgi apparatus function. So this is what is formed. So it is a tiny, simple, spherical sac-like structure which is present in the cytoplasm. Now, they are the result of packaging and processing of the Golgi apparatus. So once the Golgi apparatus is done packaging and processing this, so the lysosomes are made then. Now, a single membrane surrounds the lysosome, meaning it is surrounded by only one membrane and not more than that. It is a unit membrane structure. Lysosomes, they are rich in hydrolytic enzymes. So all those enzymes that have hydrolytic property, the lysosomes are rich in it, like hydrolases, such as lipase enzyme, protease enzyme, carbohydrates, meaning that have the property to break down substances. So those enzymes are present inside the lysosomes. So the lysosomes, they are rich in hydrolytic enzymes. Now they
Hydrolytic
are
meaning?
hydrolytic means in the presence of water, they'll cause lysis. Lysis means breakdown. So they'll break down, like lipase breaks down the lipids in the presence of water, protease break down the proteins in the presence of water, carbohydrates break down the carbohydrates in the presence of water. So it breaks it down or digests it. Got it?
Yes, sir.
Hmm. Now the lysosomes, they are active at acidic pH. So like their optimum pH is acidic, and they are also known as suicidal bags, as they do autolysis or auto digest. So whenever like there is like any imbalance in the cell or any imbalance, or you can say, there is a foreign particle, so what these lysosomes do is that they engulf that foreign particle and they burst. That is why they are known as the suicidal bags, because they do autolysis, like they do self breakdown or self digestion. Clear? Lysosomes are clear. Then comes the vacuole. Do you want to do vacuole or do you want to note down?
I think you can like teach today. I don't have the power to write, to be honest.
Can't hear you.
Ma'am, can you teach this part? It's
Yeah,
fine.
vacuoles. Yeah, I'll teach. See, now vacuoles, they are storage structures mostly found in the plant cell. Okay, and they are like they have one membrane yet again. So they are unit membrane structures, and their membrane has a name. So the membrane of the vacuole is known as tonoplast. And what happens is that it contains all the stuff that are present, or you can say that are present in the cell, but the cell does not need it immediately. So the cell stores it in the vacuole. Okay, it is responsible for the most important function is storage. So it stores the water, it stores the sap, excretory products, all of these things are stored in the vacuoles. Now, in the plant cells, the vacuoles occupy 90% of the volume of the cell. Only in the plant cell, right? They'll take up 90% of the volume. Whereas in the case of animal cell, mostly vacuoles are very temporary and no permanent vacuoles are found in animal cells. But in plant cells, permanent vacuoles are present. Now, there are different types of vacuoles, such as contractile vacuole, food vacuole, and gas vacuoles. Talking about the contractile vacuole, so this one is present in amoeba, with the help of which it can excrete out the like excrete, like it can do the process of excretion, or it can excrete out the undigested substances. So contractile vacuoles. Then comes the food vacuoles. Now these are formed inside the amoeba also. Whenever it engulfs the food particle, it forms a vacuole inside, and that vacuole is a food vacuole. Lastly, the third kind of vacuole is the gas vacuole. This will act just like those floaties that people use for swimming. They are the vacuoles present in the prokaryotic organism to provide buoyancy, so that the cell will not sink down to the bottom, rather it would float. So that is why gas vacuoles are present. Clear, students? Students, I'm saying. Clear, Rida?
Yes, ma'am.
Hmm. All right, so this was all about the vacuole and the lysosome. These are very tiny parts of the chapters. So now you can note down the rest of it.
Yes,
Hmm. And when you're done, speak a little louder so that I'll hear you, so that I can scroll.
yes, ma'am. Scroll down. Ma'am, can you scroll down? Scroll down. Now scroll down. Then scroll down. Can you scroll down? Can you scroll down? Can you scroll down? Scroll down. Maybe scroll down. Ma'am, can you scroll down? Scroll down. Ma'am, can you scroll down? Ma'am, hold on. Hold on.
Okay, any doubts up until now?
No.
Okay, do you want to continue the class or do you want to rest? Because now we're going to start a new topic, that is mitochondria. So, like, do you want to start the new topic or do you want to take a rest?
I think we'll finish the mitochondria and then end the class.
You want to finish the mitochondria? But
Yeah.
it's not tiny.
Can we do till the functions?
All right, till the functions is the last only.
Oh.
All right, so talking about the mitochondria, it is the powerhouse of the cell. We all know that. And the reason why it is called as a powerhouse of the cell, because it is responsible for the synthesis of ATP. ATP stands for adenosine
Right.
triphosphate.
Sweet.
Hmm. Adenosine
Ma'am, I have
triphosphate.
a doubt. Uh, in the prokaryotic cell, it has plasma membrane or the cell membrane, right? That is the only membrane it has, correct?
Hmm.
Apart from that, there are no other membranes, right?
Mhm. So, what is the doubt? This was the doubt.
Yeah.
Okay. Now, talking about the mitochondria, it is usually cylindrical in shape or it is sort of like a sausage shaped cell organelle. And just like how we use different stains to color different kinds of organisms or different kinds of cells. So for mitochondria, the name of the stain that we use is Janus green. We use Janus green stain to visualize it under the microscope. Now, mitochondria is a double membrane structure. It has two membranes. One is the outer membrane and one is the inner membrane. Now, the number of the mitochondria, the shape of the mitochondria, and the size of the mitochondria, it varies from cell to cell. Now, for example, if the cell is performing a lot of functions, so the number of mitochondria will obviously be more in it, and the cell which is performing less function, so the number of mitochondria will be less in it. Talking about the structure of mitochondria, so there is like an outer membrane and then there is the inner membrane. Now the inner membrane, like the outer membrane is continuous, whereas the inner membrane has infoldings that are known as cristae. The outer membrane of the mitochondria, it is smooth and continuous, whereas the inner membrane forms infoldings that are called as cristae. Talking about, we know that the ribosomes are also present in mitochondria. Apart from that, the mitochondria has its own circular DNA. So 70S ribosomes, they are present in mitochondria, and single circular double-stranded DNA is present in mitochondria. And because of this very reason, the mitochondria is known as a semi-autonomous organelle. Semi-autonomous mean it is its own regulatory body, meaning it does not rely on the nucleus for division or for protein synthesis on ribosomes. It has its own ribosome, so it can synthesize its own protein, and it has its own DNA, so it can do the process of replication also on its own.
Ma'am,
Just like, yes,
ma'am, for 70S ribosome, it's only counted for prokaryotics, right?
yeah. See, the
So,
cytoplasmic ribosomes that are there, they are 80S in the case of eukaryotes. But
yeah.
the ribosomes that are present in mitochondria and the ribosomes that are present in chloroplast, they are 70S.
Oh, so for both prokaryotes and eukaryotes.
Prokaryotes don't have mitochondria.
Oh, yeah, okay, okay, yeah, okay,
Hmm.
okay. Yes, ma'am.
So, because the mitochondria has its own ribosome and its own DNA, so it is known as semi-autonomous cell organ. Now, a set of enzymes and electron carriers are necessary for the ATP formation, and these electron carriers and the enzymes, they are present in the inner membrane itself. And this is sort of like how it looks like. It has like F naught and F1 particle that looks like a racket shape. Okay, and this is where ATP synthesis happens.
Ma'am,
Talking
what exactly is the F naught and F1?
See, it is like a part, it is known as the particle of Fernandez. What happens is that whenever the electron passes through these particles, then ATP generation takes place.
So
Okay.
basically, where the energy is there, this F naught and F1 they pass through each other. Like
No,
that.
the electrons pass through these particles and generate the energy.
Oh, okay, okay.
Now talking about the functions of the mitochondria. So the like obviously we know that it synthesizes the ATP molecule, and mitochondria is where the aerobic respiration takes place. Like there is anaerobic respiration, then there is aerobic respiration. So the aerobic respiration takes place in the mitochondria. Got it?
Yes, ma'am.
Any doubts?
No, ma'am. So, ma'am, here the gaseous exchange also takes place, correct?
No.
But it has the aerobic respiration. So,
See, aerobic respiration doesn't mean gaseous exchange. That is a different thing. Aerobic
yeah,
respiration is when
the
the
oxygen is.
carbon, yeah, in the presence of oxygen, those substances are broken down to release energy.
Yeah.
Yeah,
Okay,
that
so
happens.
inside the mitochondria, they will be broken down, and that's how the aerobic respiration is there.
Respiration is different, breathing is different.
Okay.
Got it?
Yes, ma'am.
Breathing is exchange of gases.
Mm.
Respiration is breakdown of bigger molecules into smaller molecules so that energy can be released. Got it?
So, ma'am, right now, we will have the aerobic respiration.
Yeah, we have the aerobic respiration. It goes on in the mitochondria of our body.
Ma'am, so will it also contain anaerobic respiration because, you know, it's half partial oxygen is
No.
there?
And anaerobic
I mean,
respiration,
not oxygen, sorry. Like it won't have the oxygen, right? So will it have it?
no. See, anaerobic respiration does take place in our body, but not in the mitochondria. It takes place in the muscle cells whenever there is deficiency of oxygen.
Yeah, okay.
Hmm.
Okay.
But mostly, if we are talking about the aerobic respiration, so 100% it will take place in the mitochondria. Got it?
Yes, ma'am.
Hmm. Anything else?
No, ma'am.
So mitochondria is clear.
Yes, ma'am.
Do you want to note it down?
Yes, ma'am. Ma'am, can you scroll down? Ma'am, can you scroll down?